I would like to welcome everyone attending this webinar, wherever part of the world you may be. This webinar that we organized in celebration of the UPLB Museum of Natural History's 48th founding anniversary has a lineup of speakers from different parts of the globe, all of whom are involved in preserving natural history and in museum work. Titled Museums in Leading Transformations for Species Conservation, this webinar will delve into the innovative strategies and initiatives undertaken by museums worldwide to conserve biodiversity and efforts towards sustainable conservation practices. As we utilize this online platform, UPLB MNH is honored to have been able to bring you a distinguished panel of experts in this virtual space. From leading museums and conservation organizations who will share their insights and experiences. We hope everyone takes this opportunity to explore what more we can do as museum professionals, biodiversity advocates, conservationists, educators, researchers, and students in advocating for biodiversity and our own causes. Thank you very much and have a good day to everyone.
Good afternoon. Let me just share my screen. Okay, hi. All right, good afternoon to one and all. Thank you for having me here for the webinar. Yes, as Christian has mentioned, right, I'm entomologist and the cryogenic curator. Right, so I do focus on the insects biodiversity of Singapore as well as Southeast Asia. And some of the specimens, or let's say the samples, like be it like DNA or small specimens or even blood and tissue, I will actually keep them in the cryogenic collection, right, to preserve the at least the genetic basis of your animals around us. So yes, I do go around to different places and I do search for different kind of insects, right? Basically looking at kind of insects that might be, say, not just new to the region, but also could be new to science. So on screen, what you see was actually one of my expedition trips to Sarawak. Right, one of the places that went there was the most exciting. Going deep into the jungle, spending about a week in there, right, and searching for new insects. Of course, I do encounter some of the other animals too, like the giant toad that you can see on the right side of the screen. So yeah, and no, it's not only having to do a survey or doing a search during the daytime, but also during the nighttime as well. So I can see that this is actually may look a bit scary to some, like having to be surrounded by different insects. But I would say it's also one of the entomologist's dream, right, to see count many different insects all gathered all in one spot. So this is definitely one of the fascinating points of my life. Yes, I do search for different kind of insects since I was in during my undergrad days, right? So these are the different kind of insects that I have found so far. And these were actually taken just by my phone or a normal camera, right? So I will talk more about that later. But these are the different kind of insect species that we have in Singapore, not just in our forests or like the mangroves, but also in our urban areas too. At the same time, right, there's this research work that's also being done with regards to the insect biodiversity. So some of these insects we have collected, right, we will actually take a high resolution images of all the different insects. So you can see that these kind of insects on screen right now are not the kind of insects that you usually see outside of your homes or in your gardens. These are the ones that are more specialized within the forest. And how do we go about collecting them? I will talk in the next slide. So yes, as Christian has also mentioned, also I do have a particular interest with regards to the bacteria, especially with the termites. So on the left side of the screen, you can see some of the different kinds of forest cockroaches that we have in Singapore. At the same time, on the right, you can see the different kinds of a termites. So these are not the usual kind of termites that will come and destroy your furniture or your homes. These are the ones that are actually useful with the environment, right? Like for example, the one that is dark brown in color is a one of the Nasuts termites, which actually kind of uh, feed on, say, of the lichen or the algae, not on the wood specifically. Right? And then we do have some of the termites that don't usually bite, but they will actually produce a snapping noise at the bottom of the screen. So now, I think as your team has mentioned, we are talking about the digital age, right, with regards to a natural history museum. So the museum here, we do have in a way prepared for the digital age, right? Something that has been advancing so much over the years. So how do we actually bring in digital age to regards to doing biodiversity, or in some cases, bio bleeds of the species? So I would draw only more focus on the insects itself, which is actually I'm part of. So I think we can all start off with, like, nowadays, technology has advanced so much from the days, especially we using cameras to take photos of different animals around. So especially regards to photography, right? I think some of us here may actually have taken photographs of like sceneries or buildings, right? Or even food, right? Not only just having to use, like, say, a very highly advanced camera like the one you see on the left, like the digital single lens, or nowadays even your phone are also getting more advanced, taking photographs that it can be such of good quality, such as what you see on the right, with regards to macro photography of all the different insects around us, right? This taken by the phone itself is actually more than enough to possible even tell of the genera or even the species in some cases. So I think one good example of, like, say, some of the different museums or some of the different experts and specialists around the world, they actually do use the iNaturalist in order to document the different species they have observed, right, in the environment, sometimes even their own homes also. So for example, when we talk about like the insect species of the world, iNaturalist has already shows that it's almost close to 200 species has been documented. But of course, we would have also known that there is at least maybe around 1.1 million species has been described. Though not all of them have been placed on iNaturalist. I think that is also because due to the limitations of identifying the species as well as the photography. So how is the museum, in a way, taking lessons or taking example from iNaturalist? So with regards to the biodiversity in Singapore, this is actually what we did. So I am part of this particular project or the team, what is known as the BDP, or you call it as the Biodiversity Discovery Project. So the main idea here is because we do know that we are experiencing the sixth extinction on the global scale, or what we know as the insect apocalypse. So the lead investigator Prof. Meyer wanted to find out all the different species that we have in Singapore as well as Southeast Asia before they actually disappear from our world. So it all started back in a 2010, right? Doing surveys around Singapore. What we did was actually to place up Malay traps, as what you can see on the right hand side. So you can see the Malay trap is almost as tall as I am. I'm the one wearing in the orange t-shirt. So we actually will do passive sampling, and then we place traps around Singapore and gather as many insects as possible. So what happens next is that once we have collected the insects, our idea here is to identify, right, the different possible species, and then followed by taking images of them and put it up online. And the online website that we have here for the diversity in Singapore is known as the Biodiversity of Singapore Online. So it showcase, as you can see on screen, that it's not only just insects, but also vertebrates and as well as some of the plants. So how does the project goes about doing it in preparing to document all the different species in this digital age? right? So this is actually the idea of what we have in mind, right? The goal to showcase all the different diversity. So it all started out with the Malay trapping. So we do passive sampling and we will collect the insects from the traps. So once this on the left side of the screen, as you can see, is all the different insects we have collected from just 1 week. And usually for these projects comprises of many sub-projects. Some of these projects itself, we will spend across a period of 3 months. And the longest one that we had was actually spanning across 2 years. So you can imagine on every week, let's say from different locations, up to a dozen locations, different insects have been collected. And that can actually amount to almost many million of individuals of insects to be documented. But so with that many insects itself, and then with limited manpower, how do we actually speed up the process of identifying all the different insects? So once we have collected the insects, right, we have different students, right, or sometimes taxonomists come in to help us to sort them out into the different groups. So as you can see, for one of the students here, she's actually trying to sort the different insects, right, maybe just to the orders or to the different families, as you can see on the right hand side, right. From the left hand side, the whole insect soup has been sorted out into the different insects that you see in the different families, right? Of course, we do have some of them that are much more prominent and more abundant. Some of them are much more rare or less common. So once we have identified the different kind of insects, there some of them may undergo a dissection process in preparation for the molecular work, right? And then some of them may actually we do the dry preservation, right? The usual pinning of the insects. And of course, at the end of it itself, any insects that has been identified as a possible species, we will actually do the photography, right, to produce high resolution photographs online. But I'll come into that later and how that plays a part. And last but not least is actually the DNA work. So each of these insects that we have collected and sampled, they will undergo a DNA work in order to identify them through molecular means. So this is an example of what we are doing is basically we are for every particular order or say a family of insects, we will actually put them into a 96 well plate. So each particular hole there is actually containing one particular insect, right? So what you can see actually is happening here is basically, with each well of insect, right, they will actually be given the PCR reagents, right, and then this we will do it on a mass scale. So you can imagine on each day we will actually prepare up to say 400 insects, right, then with let's say a number of manpower. We maybe let's say we have 6 people. You can imagine how many insects can be processed within 1 day. Right. This actually help us speed up the process. I think the other question that some of you may have in mind is also like, if the insect is actually too big for the 96 well, what do we do with it? Well, we will actually take parts of the insect, right? Maybe a leg or even the head. Yeah, so you basically can get an idea here on that. See, using some of the machinery, some of the advanced technology, we can actually process a lot of specimens within 1 day. Right? But this is actually having to forego with regards to the morphological identification. Yeah, so on the mass sequencing itself, right, with this 96 well plates of insects that we have around, right, you can see on the tables here, each of these particular box is actually containing a set of 95 insect specimens, right? The last well will be actually used as a non-reagent, right? There's non-insects for a negative control. So you can see there's a lot of insects that we can actually process in 1 day that we actually take up the entire lab to process thousands and thousands of specimens. So once we do the mass sequencing or the PCR of the insects, right, we of course will do get a lot of bioinformatics, a lot of data regarding to the different kind of insects. And how do we go about processing it? Well, generally on the left-hand side is actually what we'll see for each particular insect. What we did was actually to sequence the CO1 gene. So you can see there's a lot of DNA has been processed, right? So all the DNA has been processed, we will go through a clustering of the sequence insects. And then that will actually produce something what you see on the right. So what on the right you see is, well, is basically some of the different insects that you have sequenced, right? What are those colors in red? So for example, we are looking for a particular group of insects that have the same CO1 sequence, right? Through the Excel sheet and the macro function, we can actually get the identify, hey, these are the particular insects that actually have the same CO1 gene. And then from there, we can actually can more or less identify that these guys have the same molecular sequencing and should be the same species. Then from there, we can identify. So we'll go through each and every CO1 gene until we clear all the different insects. And that is actually how we cluster the different insects by a molecular means. And this is what we know as the MO2s. So molecular taxonomic units, right? So yeah, with many different clusters around, we can actually produce different cluster of insects. So of course, it can actually range from, like, say one of the singletons or one doubletons. So either a singular species or species represented by two individuals, all the way to, say, let's say 1,000 individuals actually to represent one species, right? That is actually the spectrum of sequencing all the insects. So once we have all the different clusters, right, they are all being identified by the molecular sequence, right, we will actually pick out the best representative of each particular cluster in order to identify them and to be also photographed. So for example, what you can see on screen right now, we do have a high resolution photography to photograph some of the very small insects, right, even the invertebrates. So example, like say some of the beetles, right, the Chelsea wasps, termite queen, as well as a tick. So for me, since I'm actually more of has a special interest regards to the termites, these are actually the different termite species that we have actually found in Singapore, including some of the cockroaches too. And this is actually all what we did was actually gone through the molecular sequencing in order to identify them. So molecular sequencing is also quite a way that's also useful. Is right, you only don't really fully need an expert to do the sequencing for it. Even students, right, or people who are actually new to identifying species can actually participate, because with regards to, say, the old world taxonomy of insects, trying to identify them by morphological means, you do require experts, and there's actually a limited number of them. The other advantage of with regards to molecular sequencing is actually that we can actually have to find different casts, or let's say different insects or different stages, right? From larva to your adults, right? Male or female, we can actually know which ones are actually the same species. So once we have the different possible species itself, right, we actually put up online where you do the high resolution photography, not just say the dorsal side, right, the underside, lateral, and the necessary parts that is actually used for identification morphologically. But of course, when we actually put up this online itself for other experts overseas to have a look, we don't usually just put the species name. What we will do is to intend that for each of these species, we also try to say associate them with other potential species. Like for example, if the cockroaches, who actually can be eaten by, right? So sometimes when you look, say, a particular bird species, maybe you can get to see, oh, these are some of the cockroaches that they have eaten. So of course, and then this is actually a good example of what Christian has mentioned of actually how we discover a new species of cockroach in Singapore. So because I am not really familiar with cockroach IDs, I'm more familiar with termites, right? We go through the sequencing, we get to know that this particular cockroach here, right, that has a potential ID, but we do not know the species name of it yet. So what we did was, what as I mentioned, we do the PCR, we do the clustering, and then we do the photography. And then we can share it online, right? Hoping that maybe an expert somewhere will actually encounter it itself. And in this case, that we do have Christian here who actually encountered it online. And that's how we actually collaborated into and discovering of this and naming this new species. So we actually, in a way, speed up the process of discovering all the different species around the world, right? And having to do this particular method of using high resolution photography, right? Even sometimes with your phone is also possible, especially in this digital age. We need to ramp up our speed in order to make sure that we can catch up before we lose the different species, right, due to the extinction. So, oh yes, and yeah, and that comes to the end of my presentation. I hope you have enjoyed and learned about how we go about speeding up the process of discovering potential new species, not just for the region, but also to science. Thank you.
Thank you very much, Maosheng.